Hi guys, welcome you to ADrPLEXUS INICET May 2023 Pathology Recall session. With a great happiness, I want to tell you that most of the things from the INICET this year, May 23, is from our workbook. That is okay. We are very happy to share you with the workbooks, with the screenshots of the workbooks and the questions also. I hope now you will be in a position of relaxed, a little bit relaxed, and all the questions, the calls, everything would have done this thing. So it will be like a summarizing what you've done and how is those things actually recalled and what is from our previous question. Like analysis, it will be just like an analysis of this paper. So if you ask me about this pathology paper, like any other INICET paper, pathology this year also, pathology, there are many questions came from pathology. Nearly 35 questions we could get from pathology and its related applied aspects. And to be very with a great pride, we are here to say that 32 to 33 questions are from directly from our workbook. That is workbook and also from our VSV sessions and our test and discussions, those things. Okay. So guys, actually, INICET, as always, gives importance of applied aspects of the questions, applied aspects of the subject, as well as your basic knowledge also it tests to a part, and it gives the importance of the PYQ. That is a previous year questions also like equal importance will be given on a regular basis. So let's see this pathology paper, how it was. So most of the questions, actually many questions this time they have asked from general pathology, and common topics like necrosis, apoptosis, amyloidosis, the basic questions have been asked. Like as usual, genetics has been given some weightage with genetic disorders, pedigree interpretation, and also genetic techniques. They are given the importance. And many repeated topics, even in the hematology, also like iron deficiency, anemia, and those things. Those are all the like a standard evergreen topics. Those topics have been also given equal importance. So let's see one by one the questions and discuss how this paper is. Yes. So we'll move on to the first question. Failure of expression of CD40 on the surface of B cells will lead to absence of. Yes. So they have asked us like if the CD40 molecule, if the CD40 antigen, if it is not expressed on the surface of the B cells, then what will happen? What will not happen? So lead to absence of what? Like you can see from our workbook screenshot of this picture. Yes. This B cell, this immature B cell will be expressing the CD40 ligand, the CD40 antigen on its cell surface, and the corresponding ligand will be present in the CD4 T cells. And the interaction between this B cell and the T cell is mainly mediated by CD40 and the CD40 ligand. Yes, if this interaction occurs, then only the class switching, that is, there is production of the antibodies like IgG, IgA, and IgE will be produced. And if this CD40, if it is not present, if it is not present on the B cell, then this interaction will not happen. And definitely the class switching, that is the production of the antibodies with IgG, AE, those things will not occur. So this is the thing which we discussed. And I hope you will also remember the hyper IgM syndrome, which we used to discuss. Yes, hyper IgM syndrome, it can be either because there is a mutation of CD40 ligand or there will be an absence of CD40. So there will be only production of IgM. There will not be any other class of antibodies will not be produced. There will not be any IgG, there will not be any IgA or IgE. Only IgM will be produced. So this is called as hyper IgM syndrome. Yes, this is the thing which we have discussed, and the question asked similarly the same thing along with absence. So the absence of CD40 will lead to absence of this class switching. Class switching will not occur, which will lead to hyper IgM syndrome. Hyper IgM syndrome. Yes. Moving on to the next question. Which of the following blood products storage temperature and shelf life is correctly matched? Like transmission pathology is one such area. You take either INICET or NEET exam. So definitely there will be one or two questions. If you see your last NEET exam paper also, like transmission related complications used was asked. So every paper, either NEET or INICET, they have given importance to this transmission pathology. Minimum one or two questions every year they used to ask. So similarly, this year also they've asked about, actually two questions came from blood banking. I'll show you the other question also. The first question is regarding the transfusion of this product, blood products, how much it can be, at what temperature it can be stored, and till how long, what is the maximum duration it can be stored. So it's like a sort of options were given, and the correct options given here is this: platelets can be stored at a temperature of 20 to 24 degrees centigrade for a period of 5 days. For a period of 5 days, yes. If you could see our workbook screenshot of the workbook image, you can see that either whole blood or packed red blood cells. So whole blood or packed RBCs can be stored at a temperature of +2 to +6 degrees centigrade. And for a maximum period, if you add the SAGM additive, it can be stored for a maximum period of 42 days. 42 days. And we have also discussed up to what temperature, up to what period actually you can, within what period we have to start the transfusion, and within what period you have to complete. Packed cells, so whole blood has to be started the transfusion within 30 minutes when you take it from the blood bank refrigerator, and the transfusion has to be completed within 4 hours. Within 4 hours. And coming on to platelets, as I say, 20 to 24 degrees centigrade for a period of 72 hours to 5 days, actually maximum it can be stored. And it should be started, the transmission has to be started immediately. Actually, these are the future areas, okay? Future areas where they can ask the question. So platelet transmission has to be started immediately when you take it from the blood bank refrigerator, because platelets are usually kept it in an agitator, no shaker, it will be shaking, because platelets has a tendency to clumping. So the transfusion has to be started immediately once you get it, remove it from the blood bank agitator, okay, and it has to be completed within 20 minutes. There is fresh frozen plasma and cryoprecipitate. Fresh frozen plasma and cryoprecipitate has to be transfused within a period. Actually, they can be stored at a temperature of -25 degrees centigrade or colder, and up to a period of for 1 year. They can be stored up to a period of 1 year. And this transfusion has to be, before starting the transfusion, it has to be thawed. And the transfusion has to be started within, the completed within 20 minutes. Within 20 minutes. So these are the areas actually they can ask the questions in the next topic. Okay, next exams you can expect this type of questions. Moving on to the next question. So the correct answer here is the platelets transfusion for a period of 20, stored a period for a period of 5 days at a temperature of 20 to 24 degrees centigrade. 20 to 24 degrees centigrade. Yes. So now actually we'll be moving on to the next question. A 35-year-old female presented with a foul-smelling vaginal discharge, and the microbiologist found flu cells in the smear taken. Okay, so possibly it will be a pap smear, and the microbiologist they have mentioned. So microbiologist found a flu cells. And what is the correct description of the flu cells? So they have asked us about the description of the flu cells. What are the flu cells? So flu cells actually are nothing but they are seen, as you can see from our workbook. This patient usually presents with a fishy vaginal discharge. So they have asked us about given the clinical history, the patient is having a history of fishy vaginal discharge. And we have, as we can see from our workbook, we have discussed exactly the same thing. So this kind of discharge are usually seen in Gardnerella vaginalis or bacterial vaginosis. So what is this? Basically the flu cells. Actually in pap smear, you will be seeing the squamous epithelial cells. The squamous epithelial cells. So you can see here the squamous epithelial cells. This are all the coccobacilli. This Gardnerella vaginalis is nothing but it is a coccobacilli. So along the margins of the squamous epithelial cells, this bacilli will be actually adhering to the margins of the squamous epithelial cells, giving the I say like a something like a fuzzy border, So these are called as the clue cells. Actually, in pap smear, it will be quite evident. The squamous epithelial cells with the margins actually with the small coccobacilli. So these are called as clue cells. This is the exact thing actually we have discussed. And this was the options actually seen in your given in your exams, and this thing is the correct option: epithelial cells with coccobacilli on the surface. The other things like dysplastic squamous epithelial cells or epithelial cells with perinuclear halos, these are all the coilocytes and those things. And pear-shaped organism is nothing but it is actually a trichomonas vaginalis. So it is doesn't fit. The other things are not fitting into it. So the clue cells are nothing but these are all the epithelial cells with the coccobacilli on the surface. Okay. So moving on to the next question, actually apoptosis, this was like a straightforward question. So which of the following is an anti-apoptotic gene? Anti-apoptotic gene. So if you remember our class, actually we have discussed about this proto-oncogene, that is in that we have discussed about what are the pro-apoptotic proteins and anti-apoptotic genes and the BH3 proteins. Remember, I told you to remember BCL2. That is BCL2 is actually a gene which is expressed in B cell lymphoma. It was first identified in a B cell lymphomas. So it was given the name BCL2. So BCL2 means B cell lymphomas. This gene was first identified. So lymphomas, what happened? Lymphomas, there will be proliferation of the lymphocytes. There will not be any apoptosis. The lymphocytes will be keep on proliferating. So there is no apoptosis. So they are anti-apoptotic. So please remember BCL2, BCLXL, MCL1. So all these are all anti-apoptotic. Rest of the other things like BAC, BCLXS, and the BH3 proteins like BIM, BAD, Notsa, Puma, BID, all these things are pro-apoptotic or they favors the apoptotic. And if something ends with the L, okay, if something ends with the L, please remember it is anti-apoptotic. It lowers the apoptotic, that is anti-apoptotic. So BCL2, BCLXL, and MCL1. So if you remember this concept, then this question is like a cakewalk for you. So BAC, Puma, BAGs, all these things are pro-apoptotic. And the only option which is given here, which is an anti-apoptotic, is nothing but it is MCL1. MCL1. Yes, you can see that we have discussed in actually in detail in our sessions. Coming to another very standard question and one of the every year repeated topic, that is about the iron deficiency anemia. So whether you take it in a NEET or INICET or even in the upcoming next also, actually we can expect this question, that is the iron deficiency anemia and the differences between this thalassemia, anemia of chronic disease or sideroblastic anemia, one of the evergreen topic of your exams you can take. So as we discussed enough about these things during our test and discussion and also in our BSV, so let us quickly recap very important things which can be helpful to solve this question. Yes, so in the question they have asked us which of the following can be seen in iron deficiency anemia. You can see from our workbook, that is this page, actually you remember very well we discussed this thing. So in iron deficiency anemia, obviously iron deficiency anemia, so the serum iron is going to be reduced. The serum iron is reduced, but body wants to even save a small amount of iron which is reaching the body. So the total iron binding capacity will be raised. The total iron binding capacity will be raised. Rest all the things like MCV, MCH, MCHC, transferring saturation, serum ferritin, which is the storage form of the iron, serum ferritin, which is the storage form of the iron, marrow, iron stores, iron in normoblast, everything is going to be reduced or absent. Only the total iron binding capacity alone is going to be increased in your iron deficiency anemia. Rest of the other things are going to be decreased. Yes, coming on to this options. Yes, decreased ferritin will be seen, decreased transferrin saturation also will be there, and decreased serum iron also will be there. But what you will not see is total ion binding capacity will not be decreased and it will be increased. Yes. At this juncture, just quickly recap how will you differentiate between iron deficiency anemia and anemia of chronic disease. Yes, 2 points which I told you to remember. Remember, iron deficiency anemia and anemia of chronic disease. How the total iron binding capacity will be the total iron binding capacity in iron deficiency anemia is going to be increased, and in anemia of chronic disease, it will be exactly opposite. That is, in total iron binding capacity will be reduced. Remember the other important point which I told you. Ferritin. Ferritin, which is the storage form of the iron, is going to be decreased in iron deficiency anemia, whereas in anemia of chronic disease, the ferritin is going to be increased. This is the easy and most catchy 2 points which you have to remember while differentiating between your anemia of chronic disease and the iron deficiency anemia. Yes. So as I always insist, please remember this tabular column, which will be helpful to solve you at least 1 or 2 questions in every exams. Okay. So moving on to the next question. That is, this was the very actually pathology, the general path, the first topic, that is the necrosis. This time the questions has been asked. Okay. So all of the following are true about necrosis, except. Yes. So if you remember the things which we have discussed during our necrosis, the differences between the necrosis and the apoptosis is, so necrosis, what happens? Necrosis is always pathological. Please remember that. Necrosis is always pathological, whereas apoptosis can be physiological or pathological. Yes, embryogenesis is physiological, apoptosis can occur. Pathological in many conditions, viral infections and also your DNA repair mechanism, this apoptosis occurs. So apoptosis can occur both in physiological as well as in pathological conditions. Whereas if it necrosis occurs, it is always pathological. Yes. So this option is not the correct option. So this is a wrong statement. Rest other things like cell swelling. Whenever they cell gets swollen, so, get injured, what will happen? Sodium potassium ATPase, this function will be lost, and there will be more calcium influx, sodium will be more influx, and water will be there. So hydropic swelling occurs. So the cell swells in size, whereas in apoptosis, the cell gets shrunken in size. Yes. So this is a very important point. Rupture of the cell membrane occurs in your necrosis, rupture of the cell membrane. Remember this point which I told you. The rupture of the cell membrane, so there will be leaking of the cellular contents. So whenever the cellular contents are leaked, there is always a chance of inflammation. So inflammation is present in necrosis, whereas inflammation will be absent in apoptosis. This is a basic important thing which we have to recollect here. Okay. Yes. Why? Apoptosis, the cell membrane is intact. The cell membrane is intact. It is the apoptotic bodies or like a packed structures with a well intact membrane. So the cell membrane is intact. Those cellular contents are not leaked. So there is no inflammation in apoptosis. But in necrosis, inflammation is present and a rupture of the cell membrane occurs. So swelling occurs, cell membrane rupture occurs, and inflammation is also present. So all these things can be seen in necrosis, whereas necrosis is always pathological. Apoptosis can be physiological and pathological. The same thing which we have discussed in our workbook, and you can see the 3 salient points which we have enumerated in our tabular column. The same options were asked. Yes. So let us move on to the next question. As I told that there were 2 questions asked from amyloid, 2, not 3. Actually, it's not true. 3 questions have been asked from amyloid. Let us see what are those things. Okay. So all of the following are true about amyloid deposition, except. Let us see all the options. Okay, one by one. From the last option we'll go. Amyloid is an extracellular deposited substance. Yes. The definition of amyloid, what we used to learn. Amyloid is a homogeneous extracellular material. Extracellular, it is not inside the cell. Amyloid is deposited outside the cell. And what is the electron microscopic picture of your amyloid? It is a beta plated sheath configuration. Remember the thing I told. It is because of this configuration, beta plated configuration only. The other name for amyloidosis is beta fibrillosis. Yes, the other name for amyloidosis is beta fibrillosis. It is because of this beta platter shape configuration. Yes, we have two types of mainly the amyloid. And if you see from our workbook, we have discussed these things. Actually, amyloid. The primary amyloidosis, it is mainly the AL, amyloid light chain. You can see from a screenshot from our workbook, we have discussed primary amyloidosis is AL and secondary amyloidosis is AA. Secondary amyloidosis is due to secondary to some any chronic inflammation, rheumatoid arthritis or tuberculosis or bronchitis. So in secondary to this chronic inflammation, you will be getting this amyloid associated protein. So let us see in the option what they are given. So they have given amyloid AL is a secondary type of amyloidosis. No, AL is not a secondary type of amyloidosis. A is the actually AL is a primary type of amyloidosis. It is actually a primary type of amyloidosis. So this is a wrong option. This is a wrong option. Moving on to the next option, that is a composition of the amyloid is starch. Actually, previously they thought that it was a starch-like substance, but please remember the definition of amyloidosis itself says that it is an extracellular proteinaceous material. Yes, so amyloid composition is protein. It is not a starch. Okay, so amyloid is like a misfolded proteins, abnormally the degradation of the proteins which is lost. Okay, so amyloid is a proteinaceous substance and it is not a starch. So this option is also a wrong option. This is also a wrong option. Okay, so there are two right and two wrong options in this question. Okay. So moving on to the next question. Actually, they have given a history of an elderly male with a renal disease was given. History of a renal disease was given, and obviously it will be understood like a chronic renal failure. The patient had gone for renal transplantation. I suppose there was a given history of hemodialysis. History was given in your question. So in that case, chronic renal failure and also with hemodialysis, what type of amyloidosis you will think? Yes, you can see from our workbook discussion, actually we have discussed about these things. Hemodialysis associated amyloidosis is nothing but our A beta 2 microglobulin. Yes, in the table of column, actually, if you would have read this table of column, you could have answered the two questions from the amyloid. Yes, so two questions directly from this table actually has come. So the most likely type of amyloid which is deposited in a chronic renal failure in a patient with a long-standing hemodialysis patient is A beta 2 microglobulin. And you remember, A beta is nothing but it is actually present in the amyloid which is present in the Alzheimer's disease. Yes, you remember this is A beta is Alzheimer's disease. Alzheimer's disease. And AA we have just discussed secondary to chronic inflammation, and AL is amyloid light chain in the multiple myeloma. Multiple myeloma. You could see this table of column will be helping you to answer the two questions from amyloid. Okay, and moving on to the next question. Which of the following immunofluorescence pattern is shown in the image below? Yes, this time they have given you a immunofluorescence image, and this is actually a quite area of a high yield topic in your both for your NEET as well as for your INICET exams. So immunofluorescence patterns in this connective tissue disorders, like in the this ANA patterns and this antibodies pattern is one of the a very repeat topic. So you could see from our discussion, actually in our workbook, we have discussed about the main common four topics, okay, four patterns of the immunofluorescence pattern. Yes, one is the diffuse pattern in which actually the antibodies showing positivity will be staining the entire cell. That is the diffuse pattern. And if you could see the other pattern, it is the speckled pattern, like this type of speckles. This type of speckles you can see. So you call this as a speckled pattern. The third pattern which we have seen, only around the nucleolus alone, the antibody showing the staining, that is called as the nucleolar pattern. And the last pattern which we have discussed, like only in the centromeric region, only in the dot, like only in the centromere, only in the dot pattern of staining, that is called as the centromeric pattern. And what we could get from the students and their response, Actually, it is the centromeric pattern of the immunofluorescence, which was given in your exams. So, the similar to this type of image was given in your exams. And actually, there were some students were saying that the image was not opening properly. So, but what we could see from the majority of the students, they told that this type of image was given. So, we'll conclude that it will be a centromeric pattern of immunofluorescence. Okay, pattern. So, again, in the next exam, I hope we will be all will be settling in your upcoming means this INICET and thing itself. So students who are preparing for the who are freshly joined and preparing for the upcoming NEET or next exam. So please beware that this is a area of high yield topic. Okay, next year also there will be a chance of MCQs asked from this. Okay. So another very important and repeat topic, and last NEET exam also, this question actually, similar sort of questions actually came. So what is this question? Actually, a 70-year-old man presented with enlargement of multiple lymph nodes. So the patient, elderly male, so elderly male presented with multiple lymph nodes and total count and 65,000 and 65,000 was the total count, and they have given you the peripheral smear findings, and this peripheral smear finding was similar to like this, and where you can see like lot of mature lymphocytes, and what are these cells? These are all the smudge cells or the basket cells. Okay, smudge cells or the basket cells. Okay, so with this image actually we could make a diagnosis of CLL. Yes, CLL, elderly male, age is an important clue in the thing, and full of mature small lymphocytes. It is none other than it is our CLL. Okay, so they have asked us the what is the investigation is needed to confirm from the diagnosis. Yes, so how will you confirm a case of CLL? Yes, you can make a diagnosis from even from a peripheral smear, but for confirmation, what is needed? What you can see from here, it is mainly actually this CLL is expressing the very classical feature of the CLL immunohistochemistry. This IPT immunophenotyping is nothing but CD5 and CD23 positive. Yes, the CLL is positive for CD5 and CD23, which is a very important characteristic. And out of this investigations which is given, which one can tell you like CD5 and CD23, the expression of the markers, expression of the proteins, especially in the leukemias, which investigation will tell you? Yes, it is the flow cytometry. Flow cytometry only will tell you whether the antigen is expressed or antigen is not expressed. Okay, so fish can tell you about the chromosomal numerical abnormalities or any fusion product. If it would have been a case of CML, if it would have been a case of CML, like Philadelphia chromosome, how will you confirm? BCR ABL gene fusion, 922 fusion, how will you confirm? Now, then the answer would have been fish. But they are asking you about the expression of a protein molecule. So here the answer is flow cytometry. Okay. So bone marrow biopsy will not tell you the confirmatory for the CLL, and the answer here is the flow cytometry. Yes. So you can see that we have detailedly discussed about the CLL in our workbook also. And another important thing which I always used to tell you, like when we discuss about the genetics, minimum one investigation, actually one recent advances of genetic technique will be asked in any exam, NEET or your INICET exam. And this time also they have asked you, okay, which of the following investigation is used to detect alteration in the DNA sequence, alteration in the DNA sequence? Yes, as we could see from our discussion, actually, these are all the PCR-based methods which can detect the sequence alteration. Like we have discussed about the Sanger sequencing, pyro sequencing, restriction fragment analysis, length analysis, amplicon length analysis, real-time PCR. Actually, this pyro sequencing will be like can detect only the mainly this alteration in the DNA sequencing. Yes, pyro sequencing, RFLP. But actually, flow cytometry, as I just told you, that flow cytometry is just a presence or absence of expression of an antigen. So it will not detect you about the change in the DNA sequence. So flow cytometry will not be showing the detect the alteration in the DNA sequence. Similarly, fish also the DNA sequence alteration will not show. But your pyro sequencing and RFLP will definitely show you that whether the DNA sequence is altered or it is like the same thing. So these two investigations will be the correct option in this question. Okay. So moving on to the next question. Identify the component highlighted by the stain shown in the image. Yes, similar sort of image was given in your exam. So it is something like a blood vessel. So you can see this is the blood vessel. And what you can see from the blood vessel is like this pink color deposited material around the blood vessel. So this is not your normal hematoxylin and eosin stain. What is this? Especially when you see this type of very pinkish color material, salmon pink they used to say. And it will be deposited under the around the blood vessel. So yes, yes, many people have correctly said that it is like a Congo red. Yes, so Congo red, all of us know it is the staining for the amyloid. Yes, so this is nothing but amyloid deposition, which was deposited around the blood vessel and which is highlighted by our Congo red as a salmon pink color. Yes, so see our workbook image. We have discussed the same thing and a similar sort of image actually we have given in our workbook, like a Congo red showing this pinkish color material, salmon pink color material. Yes. So this Congo red ordinary microscope, it will be showing this salmon pink color, and whereas in a polarized microscopy, this Congo red will show, yes, the characteristic apple green birefringence. And also you will remember this. What is the best side to demonstrate amyloid? Yes, it is the abdominal fat aspirate. The other sides, remember, it is like the gum and gingiva and the rectum or the other sides. But abdominal fat aspirate is the most common site for aspiration of the demonstration for the amyloid. Yes. So as I told that amyloid, actually you got three questions this time. You got three questions this time. Okay. So moving on to the next question, which of the following is the most common hereditary cause of hypercoagulability? This is a point which we have discussed very humpty number of times during our discussion and test and discussion and BSV also. So there are many causes of hereditary cause of hypercoagulability, and if you can take all these causes like protein C deficiency, protein S deficiency, and your factor 5 laden mutation, antithrombin deficiency, all these things can cause a hypercoagulability. But if we can take what is the most common cause, it is the factor 5 laden mutation. What is this factor 5 laden mutation? And what is this protein C does? Remember the discussion actually we had in our workbook. Yes. So this thing, if you could have recollected this box which was given in our workbook, you could have solved this MCQ. Yes. So protein C is nothing but actually this protein cleaves or destroys or degrades the factor 5 and factor 10. Okay, factor 8, sorry, factor 8. So you can see that in factor when this protein C and protein S are deficient, so what happens? These factors are not degraded. These factors are not degraded, so their level increases. So protein C deficiency, protein S deficiency causes a hypercoagulability. Hypercoagulability because the clotting factors 5 and 8 levels are increased. Factor 5 and 8 levels are increased. See here. Factor 5 and 8 levels are increased. But what is this factor 5 laden mutation? That is factor 5 is showing some resistance. That is when even protein C try to degrade this factor 5 and 8. Now the factor 5 is showing some sort of resistance that it is not degraded by the protein C. So factor 5 level increases. Factor 5 undergoes mutation, and factor 5 is resistant to the action of protein C. And I hope you remember the thing, no? Some of you asked why it is called Leiden. I told it is the name of a city in Netherlands, actually, from where this mutation was first identified. So this factor 5 Leiden mutation is the most common genetic cause of hypercoagulability. Okay. So moving on to the next question. A female presented with a skin thickening and a pain in the fingers on exposure to cold. Yes. So skin thickening tells you that they can give you a suspicion of your scleroderma, skin thickening, and fingers on exposure to cold and muscle. Cold, it is something like a Raynaud phenomenon. On exposure to cold, the fingers actually pain is same. And also muscle weakness tells you actually it is like a systemic symptom. So actually they have given a thing like this patient is positive for ANA and SCL70. And they have asked us, like, what are the other antibody which can be positive in this patient? Like, if you could see from our discussion, actually, this scleroderma is of two types. That is the limited type and the diffuse type. Actually, limited means, actually, there will be only the distal extremities and face will be involved, whereas in the diffuse type, actually, there will be proximal extremities, trunk, and distal extremities, face, and visceral involvement also will be there. So diffuse, visceral involvement, extremities, face, everything is involved. So this is limited, this is diffused. So you can see this in diffuse scleroderma, you can see anti-SCL 70 will be positive. Anti-SCL 70 will be positive. Whereas in limited, it is anti-centromere will be positive. So you can see these are all the options actually given in your exams. Anti-centromere, anti-UTRNP. They have told you that the patient is SCL 70. And also they are told that it is a muscle weakness. So this muscle weakness are more prominent in your diffuse type, and also you can see that anti-SCL 70 is positive. So all this favors a diffuse scleroderma. You can ask me, sir, that patient actually has a pain on exposure to cold. Yes, this can be a part of your crest syndrome, like calcinosis, Raynaud phenomenon, esophageal dysmotility, sclerodactyly telangiectasia. Yes, crest syndrome can be seen both in limited type as well as in your diffuse type. Yes, but it is more commonly seen in limited type and less commonly seen in diffuse scleroderma, but can be seen in diffuse scleroderma. Okay, so moving on to the what are the antibodies which can be present in this scleroderma. So other antibodies, if you can see from our table column again from the workbook we have discussed, actually crest syndrome alone, we would have chosen the option anti-centromere, but we have now made a diagnosis of a diffuse scleroderma. Diffuse scleroderma. Why we have made a diagnosis of diffuse scleroderma? Because the examiner has given us that this patient is positive for this anti-SCL 70. Yes, moving on to this table column from our workbook, like we could see here, directly it is given as diffuse scleroderma. It is positive for fibrillary, which is otherwise called as anti-U3-RNP. Yes, so this table column from our workbook directly gives you the answer. That is, this patient is positive also will be for anti-U3-RNP. So this will be the correct option for this question. Okay. Yes. And one of the most favorite and repeat question which is asked in almost every exam, I used to say there will not be any paper without a question on a vasculitis. Whether you take a NEET exam or next or INICET, anything, there will not be a question paper without your vasculitis. Yes, this time also the one of the examiner's favorite vasculitis they have given. And what is this thing? A child presenting with this kind of tongue. Yes, many of you would have clearly diagnosed this as a case of a strawberry tongue, and you would have been made a diagnosis of Kawasaki disease. Kawasaki disease. Yes. So, and also what is the other picture actually I could get from the students is like the patient had a non-purulent conjunctivitis. Image of a non-purulent conjunctivitis was given. So the patient with a strawberry tongue and a non-purulent conjunctivitis, and I hope you remember the other criteria for that Kawasaki disease. Remember the cream criteria? Yes, we discussed from our workbook the same cream criteria. Conjunctivitis, rash, and edema or erythema of the hands and foot, and cervical adenopathy, and mucosal involvement. So this constitutes the cream criteria. Along with the fever, this constitutes the cream criteria for Kawasaki disease. And they have asked us about the what is the management, appropriate management is. So the main dreadful complication in Kawasaki disease is, especially in the children, it is like the coronary artery involvement. Coronary artery involvement. So coronary artery involvement, coronary aneurysm can even cause myocardial infarction in a child. Remember this point which I was insisting you? Myocardial infarction is the in a child. You have to think of Kawasaki disease. Okay. So what the treatment first will be aimed is they will be giving the IV immunoglobulins. Okay, gamma globulins they will be giving. Well, the thing is to reduce the incidence of the coronary damage. Okay, so IV immunoglobulin will be the first line of management or the appropriate management to prevent the complication, because the complications are going to be a coronary artery involvement and myocardial infarction. To prevent the coronary artery damage, they will be giving you the IV immunoglobulin. Yes, so you could see the cream criteria applied here. And this is also the one of the repeat topic, as I told in the beginning. This INICET exams had a lot of the previous year questions. Yes, so previous year questions, guys, please remember That is the basic thing. In and around the same questions only they will be asking you, same topic only they will be asking you the 80% of the question. So if you're very thorough in your PYQs, definitely you will have an extra edge in the exam. Yes. So let us see the next question, which is also a very standard repeat question. They have given you the pattern of inheritance and in a pedigree format, and they have asked us what is the pattern of inheritance. Yes. So this is the pattern in which actually what are the things which I told you to remember when you want to interpret a pedigree. First, you have to see whether males and females or both are genders are affected or only one gender is affected. Yes. Now keep on analyzing on this pedigree. Which gender is affected? Both females as well as males are affected. Yes. So then what is the other point which I used to tell? Please check whether whether the disease occurs in every generation or whether there is a skipping of generation. Yes, you can see here the first generation also people are affected, and in the second generation also people are affected, in the third generation also the people are affected. So there is no skipping of generation, no skipping of generation. So both males and females are affected. So we are ruling out the X-linked recessive disorders, and there is no skipping of generation occurs. So we are ruling out autosomal recessive. So in autosomal recessive, there is skipping of generations will be occurring, and in X-linked recessive, there will be females will be carriers and males will be affected. So with this thing, we are ruling out the things, and we are left out with autosomal dominant and X-linked dominant. Now see the pattern of inheritance. See, a male is transmitting the disease to both the son and the daughter. Okay, see, this male is transmitting the disease to both the son and the daughter. And also see the female. See this female is transmitting the disease to both the son and the daughter. So male transmitting the disease to both the son and daughter tells you that actually it is not a X-linked disorder. If it would have been an X-linked disorder, what will happen? X-linked disorder. Males will transmit the disease only to the daughters. Yes, you remember the point. Why? Because males are XY. Males are XY. So a father gives X chromosome only to the daughter, only to the daughter. So a father gives Y chromosome to the son. So a father transmitting the disease only to the daughter means it is a dominant X-linked dominant disorder. But here the father is transmitting the disease both to the son and the daughter. So we are ruling out X-linked dominant, and it is the autosomal dominant. Autosomal dominant. Okay. So you can see this. The same description which we have discussed in our workbook, and also we have discussed about the list of the autosomal dominant disorders also. We have discussed all the patterns of inheritance. Okay. So moving on to the next question, which of the following ion flow is affected in cystic fibrosis? This is actually like a very actually big, easiest question which I could say. Okay, so cystic fibrosis we have enough discussed in our workbook, and you could see from this what happens in our, we have discussed in our workbook what happens in a sweat duct and what happens in the other exocrine glands. Yes? So in a lumen of the sweat duct, normally what happens? This CFTR, actually the chloride, there will be inflow will be there, and ENAC, there will be inflow of the sodium. But what happens in cystic fibrosis, it is mainly the chloride channel gets shut down, and so there is no influx. Okay, there is no influx of the chloride into the epithelial cells of the sweat duct. So there will be more chloride going to be remaining in the extracellular. So increased chloride content in the cystic fibrosis is because the chloride channels are mainly affected. It is mainly the chloride channels are affected. And please remember, here they are given as the option. So mainly in cystic fibrosis, especially in the sweat glands, actually the chloride channels are affected. Okay. Which of the following cell is shown in the image below? Yes, they are given, if the examiner has given you some black and white image, please think that it is usually an electron microscopic. There are some only 5 to 6 images electron microscopic are very standard and characteristic will be asked in your exam. So one is, you remember we discussed about this minimal change disease, flattening of the foot process, and also remember the Alport syndrome. basket wave basement membrane, okay, in Alport syndrome. And also the electron microscopic picture of our Langerhans cell histiocytosis, that Bilbeck granules, tennis racket shape. And another characteristic picture of, usually you will be getting these Gaucher cells, that zebra bodies, and also your Niemann-Pick disease, that zebra bodies you will be seeing, and Gaucher cells, that lysosomes with the stacked lipid bilayer. So all these things are the images, and definitely this is not a Gaucher cell. And what you could see from this cell is electron microscopy. You can see this is a nucleus, and you can see from the cytoplasm there is projection of the hairy projections. Yes, this is a very characteristic thing you could see here. The cytoplasm was of the cell showing the hairy projections. So this is a case of hairy cell. Hairy cell. Hope you remember this discussion we had during our hairy cell leukemia. And this hairy projections can be better appreciated or actually made up of ribosomal lamellar complex, and they are better appreciated in a phase contrast microscope. Yes. And I also will be definitely you will be remembering the markers present in the hairy cell leukemia. That is a quite number of markers that is very characteristic of your hairy cell leukemia: CD11C, CD25, and CD103, and CD123, and annexin A1. Yes. And also they are TRAP positive, tartrate resistant alkaline phosphatase positive. And I also hope you remember hairy cell leukemia, honeycomb appearance of fried egg appearance on the biopsy. Aspirate, you can see the hairy projections, whereas if you take biopsy, bone marrow biopsy, trephine biopsy, you could see this fried egg appearance or honeycomb appearance, very characteristic of your hairy cell leukemia. Okay. And moving on to the next question. Which of the following are true about the BRCA1 gene? Yes, BRCA1 gene is nothing but actually very, very important topic, and we discussed quite when we discussed about the carcinoma breast and the ovarian cancers. Yes, so this is nothing but this is a tumor suppressor gene. BRCA1 is a tumor suppressor gene, and we could see from our discussion it is a ovarian cancer. It is apart from the breast cancer, it is also implicated in the ovarian cancers. Yes, and this is present in chromosome 17, and they are positive mainly in this triple negative cancers. BRCA1, triple negative cancer, they are poorly differentiated cancers. So coming to the option, yes, BRCA1 gene is a tumor suppressor gene. Like your P53 and the retinoblastoma RB gene, BRCA1 gene is also a tumor suppressor gene. BRCA1 is associated with ovarian cancer, and definitely this BRCA1 also has some relationship with your Fanconi anemia. Fanconi anemia. And but colonic cancer, it is not that much associated. So, like out of all the other options given, the best option to choose actually which of the following are true about BRCA1 gene. So the first option, tumor suppressor gene, is a true one, and ovarian cancer is a true one, and Fanconi anemia is a true one. But what is not true is that it is not associated with the colonic cancer. Okay. So moving on to the next question. One of the very standard and very interesting questions is I could see like most of the questions actually like whatever the things we have repeated and regularly stressed, it's like a standard repeat thing. So I'm again saying, so repeat topics or previous year questions. So who has seen all the previous year questions and done well in this previous year questions definitely will get a good score in this INICET exams, because most of the topics were repeat questions. I could see only few questions actually from a new topic. That too, I'll not say it's totally new topic, but from a tangential area from the high topics. Okay. So again, one such question is the slapped cheek appearance, which may lead to which hematological abnormality? Yes. So all of you will remember that slapped cheek appearance. You could see from our test and discussion and workbook, they slapped the cheek. The exact picture actually we have given in our workbook, that a slapped cheek appearance, it is actually seen in parvovirus B19. What is this parvovirus B19 causes? It is causes this fifth disease or erythema infectiosum, and gives you that slapped cheek appearance. Yes, you remember this parvovirus affects P4P. I told you. Parvovirus affects this affects the proerythroblast, and mainly it will be causing this intranuclear inclusions at the proerythroblast. And there will be also that you can see here from our discussion, we had that intranuclear inclusion And the cytoplasm will be showing some projections which will look like a dog ear appearance. Yes, so dog ear appearance as well as this intranuclear inclusion characterizes this proerythroblastic inclusion of this parvovirus. And coming on to the option, which hematological abnormality it will lead to? Yes, pure red cell aplasia. Pure red cell aplasia. Remember, this parvovirus can cause aplastic crisis in the background of a hemolytic anemia. Yes, I repeat it. Parvovirus can cause aplastic crisis in case of a hemolytic anemia, or especially in sickle cell anemia. But commonly it is associated with this pure red cell aplasia. What other disorder will come to your mind if you hear this pure red cell aplasia? Yes, it is thymoma. Yes, thymoma is also associated. Myasthenia gravis thymoma is also associated with this pure red cell aplasia. So the correct option here, the slab cheek appearance, actually will the parvovirus infection will cause this pure red cell aplasia. Pure red cell aplasia. Yes. Moving on to the next question. An elderly male with a history of diabetes was on immunosuppressive drugs. Biopsy of the lesion showed oval budding yeast cells with pseudohyphae. What is the most likely diagnosis? What is the most likely diagnosis? This is the standard question we discussed from our workbook, and you can see the image. Actually, we have seen exactly the same image we have given you. The budding yeast form, yes, budding yeast form and pseudohyphae form, pseudohyphae forms. Remember this candida, I used to tell that the peculiar appearance of the candida in your pap smear is like a shish kebab appearance. What is this? The fungal filaments, and the epithelial cells will be hanging on the hyphae of the fungal filaments. The epithelial, squamous epithelial cells, they will be hanging like this thing. And this appearance is called the shish kebab appearance. And candida, always an infection which you have to think in a diabetic oral thrush, okay, curdy white discharge. You have to think of this candidal infection. Further, the patient is also an immunosuppressive drugs. So they have given you budding yeast and pseudohyphae. What else? So definitely this is a diagnosis of a candida. Okay. So moving on to the next question, which is like a straightforward MCQ with match the following sort of things. Very easy one. Kaplan syndrome. Yes, Humpty number of times we have heard about this Kaplan syndrome. Yes, Kaplan syndrome. Definitely you will be thinking of cold worker pneumoconiosis. Cold worker pneumoconiosis plus rheumatoid arthritis. That syndrome is called as the Kaplan syndrome. So mesothelioma, which is actually associated with this asbestosis exposure, which is a specific tumor in asbestosis. Mesothelioma will be causing this pleural effusion. And coming to this asbestosis, remember, most of the pneumoconiosis actually will cause actually a superior upper lobe involvement, whereas asbestosis will cause lower lobe involvement. Asbestosis will cause lower lobe involvement, whereas silicosis and all will cause the upper lobe involvement. And silicosis is the one which is mostly related to this crazy pavement pattern. So this is the option, and definitely this pneumoconiosis chapter would have been a cakewalk for you, as we discussed a lot during our workbook discussion. Okay. So moving on to the next question. Which of the following is required by the thymus for the regulation of T cells? Yes, this is a question which we discussed when we discussed immunology about the autoimmunity central tolerance. When we discussed about the central tolerance with this T cells, we have discussed this thing. So remember from our workbook this page. Yes, so we have discussed about what is the central tolerance in which the self-reactive T cells, they're doing the processing. Actually, there are some T cells and B cells which will react against the self-antigen. So body has to remove these T cells and B cells. So what is the gene which is responsible for this? The gene which is responsible for this is AIRE. Remember the same page we have discussed during our workbook. AIRE. What is this A for AIRE stands for? I told you that it is autoimmune regulator protein. Autoimmune regulator protein. So the thing asked is yes. So this is actually the thymus for the regulation of T cells. You need this AIRE gene. AIRE gene. So what will happen if AIRE gene is not there? That is, there is no one to regulate the T cells. So T cells, self-reactive T cells will be proliferating. So there will be autoimmune disorders all over the body, especially it will be affecting the endocrine organs. So what is this disorder called as? autoimmune polyendocrineopathy. Autoimmune polyendocrineopathy. So multiple endocrine disorders will show this antibodies targeting the antigen, that is the tissue. So mutation in AIRE will cause autoimmune polyendocrineopathy. The same exact question they have asked from our workbook was asked in the exam. Very happy to share with you. Yes, this is the question which I insisted doing when I took genetics, and I told you that from genetics is a highly topic in your INICET exam, and I told you that minimum 2 to 3 questions will be asked from genetics, and you could see from this discussion of the questions itself, some 3 to 4 questions were asked in genetics. I have a few more questions in genetics to show to you. Okay, so what is the karyotyping? It is the like arrangement of the chromosomes in a descending order. So how will you take this? How will you collect the samples? Yes, the blood samples, you remember when I discussed even the back container also, I told you that usually if you want to connect this chromosomal studies, you have to collect in a heparinized vac container. Yes, so blood sample has to be collected with a heparinized vac syringe, and it has to be incubated for 37 degrees centigrade for 3 days. And what next you have to do? You have to add colchicine, the same thing which we have discussed in our workbook. We have studied about like what is karyotyping, what are the blood samples, and how will you do a metaphase arrest. Remember the colchicine also we have discussed. Okay, so then we have to prepare the cell colonies. And what is the regular staining which we will do in a karyotyping? Remember the thing which we discussed during the banding technique, which is the commonest banding or standard banding technique. It is the G banding. That is nothing but Jimsa banding. Yes. So remember G banding, Q banding, C banding, R banding, all these banding techniques which we have discussed in our workbook. Okay, these banding techniques we have discussed. So finally only you will be doing this banding, and then you will be studying the chromosomes. So these are the sequential steps in the karyotyping. Moving on to the next question. A patient presented with features of colonic cancer, and which of the following investigations is usually not indicated in the management of the colonic cancer? Yes, so CEA is actually a tumor marker for colonic cancer. Colonoscopy will be definitely useful in the management and the diagnosis of the colonic cancer. CT scan to know the staging of the tumor, and usually the CA 19.9 is a tumor marker for the pancreatic cancer. So CA 19.9 is a tumor marker for the pancreatic cancer. So it is not actually, will not play a much role in the management of the colonic cancer. Okay. So this is the 25th question. And coming to the 26th question, which we discussed now is the match the following. Again, I told you, the genetics question they have asked. So it is like one of the standard questions and very easy questions. Yes. So trisomy 13, all of you will be saying that trisomy 13, Patau syndrome. I told Patau, P, 13, 13. So 13, Patau syndrome. I told you to remember like that. Okay. Then what I told you about 18, E for 18. So E for it is Edwards syndrome. And remember the trinucleotide repeat disorder which we discussed. Yes, I used to tell this trinucleotide repeat disorders are very, very important thing. And Huntington disease is CAG repeat. Okay, it is in the coding region. Remember, most of the things are in the non-coding regions, and Huntington disease is the trinucleotide repeat in the coding region. Yes, we all discussed about this Patau syndrome, Edwards syndrome, and trinucleotide repeat in our workbook. So this question will be like a cakewalk for all of you. Yes. So trinucleotide repeat disorder is a Huntington disease and point mutation. Yes, you all remember. Okay, glutamic acid is replaced by valine. That is actually occurring because of a point mutation, sixth position of the globin chain. So point mutation. So this will be the correct option. Trisomy 13, Patau syndrome, trisomy 18, Edward, trinucleotide repeat disorder. It is a Huntington disease, and point mutation is sickle cell anemia. Yes. And the fungus actually, which is there shown in the image, the same sort of fungus is shown. And as you could see from our workbook, the same thing which we have discussed, mainly 2 types of morphology you have to remember. The fungus, which is having a acute angle branching, the same thing, acute angle branching. See this, acute angle branching, and with the septate. See this septate. So a fungus with the septate type A and acute angle branching, you will be thinking of aspergillosis. And what is the other in contrasting fungus you will remember? The contrasting fungus you will be remembering is the broad aseptate fungus, which are branching at a 90 degree. Okay, which are branching at a 90 degree. As you could see here, 90-degree branching is there. So this is the mucormycosis, that is the black fungus. All this fungus we have there. Not that black fungus is that a mucormycosis. So in our questions, actually, it was mainly the aspergillus. See this. This is like a acute angle branching, and you could see the lot of septate in this, So septate acute angle branching will make you a diagnosis of aspergillosis. Moving on to the next question. It was like an assertion and reasoning type of question. And the patient presented with a flaccid bulla and a suprabasal split. And there was a confusion regarding the options given in this thing, in this question. Reasoning, actually, some of you told that it is a row of tombstone appearance. So I'll tell you the pathology, then we can analyze like what is right and what is wrong. So in pemphigus vulgaris, it is basically a suprabasal split. Suprabasal means what? Stratum basal will be there. This is the stratum basal will be there, and the bulla will be just above the stratum basal. Okay. And it is intraepidermal. That means what? It is inside the epidermis. So it is intraepidermal suprabasal. So only the basal cells alone are there. No? So it is called as a tombstone appearance. So what you will see inside the bulla? Inside the bulla, it is mainly the desmoglins, all these thing, connective things are lost. So the cells will get separated. Epithelial cells will get separated, and they will be assuming a round morphology. Okay, round morphology, and these cells are called as the acantholytic cells. Okay, and this row of tombstone appearance you can see in your ordinary microscopy. Okay, in your light microscopy, you will be seeing this tombstone appearance. But what you could see in your immunofluorescence is the fishnet appearance. In the fishnet appearance of the immunofluorescence, all these things are about the in pemphigus vulgaris. So a patient with suprabasal split is a correct one. That is a correct option. And though tombstone appearance, if it would have been given in a light microscopic picture, it is right. But this, because of acantholysis suprabasal split only, this row of tombstone appearance occurs in a light microscopy. Whereas if it would have been asked in immunofluorescence, it would have been a fishnet appearance. There was a similar question which was asked in your from this bullous disorders. A female presenting with a bullous lesions, and they are given this IF showed is IgG and C3 deposits. What is the most likely diagnosis? The image, actually, most of you were telling that it is a linear pattern of immunofluorescence. Yes, see this. This is a linear pattern. This is a linear pattern of immunofluorescence signal you are getting here. So usually this thing, you will be having subepidermal bulla. You have mainly two diseases, bullous pemphigoid and dermatitis herpetiformis. Both will be having a subepidermal. Below the epidermis only the clumping of the bulla occurs. So what is the main difference, or how will you differentiate this? Please remember, if it would have been a dermatitis herpetiformis, there will be a IgA deposits, which I told you in your workbook-based discussion. Yes, IgA deposits, and if the tip will have neutrophils, you will be thinking of a dermatitis herpetiformis. But what they have said, it is IgG and C3 deposits. So we are ruling out your dermatitis herpetiformis, because dermatitis herpetiformis is usually IgA. And we are coming to a conclusion of it with the linear pattern of immunofluorescence and also with your IgG and C3 deposits. We are coming to a conclusion of bullous pemphigoid. Yes. So this is the 29th question. And moving on to the 30th question, which we are getting from pathology. That is, which is a correct order of drawing of the blood samples. Yes, we could remember from our workbook about the vacutiners, we have discussed a lot, and which order we could see that removal of the, we will collect the blood. First, when you want to collect the samples from your culture tube, you want to collect the samples for culture. The culture tube will be the first one to be drawn, and followed which you will be having a, you will be drawing blood from the light blue topped one. Okay, light blue topped one. That for the coagulation studies. That is the citrated sample. Yes, you could see from here that blue topped one or the citrated sample, the coagulation studies, because you will get a, after culture immediately you have to draw this culture sample. Means coagulation studies. Then the order is like you can remember your signal. Okay, traffic signal. Traffic signal, what you'll remember? First red, then yellow, then green. That is the traffic signal you will be remembering. Yes. So then the first, after sodium citrate, you will be collecting in a red tube. What you will be having in the red tube, you will not have any anticoagulant, and you will be collecting samples for the serological test. Serological test. Then only you will be having this golden color one, which is also will not have anything, and will be collecting blood for the serological studies. Okay, so red followed by yellow. And what is the next one? Green signal, traffic light, green signal. Heparin tube, light green. Yes, few minutes back also I was telling you heparin, what you will be doing for chromosomal studies, for chromosomal genetic studies. You can see G for G, genetic studies, green color vacutainer. G for G, I told you to remember. Yes, G, genetic studies, green color vacutainer, which contains the heparin. Yes, the karyotyping I told, no, heparinized tube blood samples has to be collected. The next one is our classical pathology one. That is the CBC, all this thing that is the last one. Before last only we have to collect this ADTA sample, ethylene diamine tetraacetic acid. All the CBC and routine blood investigation. Then the last one, you will be collecting the gray colored one, that is for the blood glucose levels, which contains sodium fluoride to prevent the glycolysis. It will stop the glycolysis. Okay, sodium fluoride inhibits the hemolysis. So all these things we have discussed, but the order you have to remember: culture tube, then blue topped one which contains a sodium citrate. Then remember your traffic signal, that is the red, yellow, and your heparin. Then comes the pathological one, that is the ADTA, and the last one is the sodium fluoride. This is the order you have to remember. Okay. Now, the 31st question, I told you that one more question was asked from your transfusion pathology. That is a massive blood transfusion. Okay, massive blood transfusion. When the blood volume is transfused, equal blood volume is transfused within a span of 24 hours, you call it as a massive blood transfusion. So, a lot of electrolyte imbalances can occur in case of your massive blood transfusion. The major one is the hyperkalemia. Okay, hyperkalemia. Usually, hypokalemia will not occur. Excess blood transfusion, all the things cells will be releasing the more of potassium. So hypokalemia will be there. Okay. And the oxalate in the anticoagulant can precipitate the calcium. So hypocalcemia will be there. And hypothermia is a dreadful complication of massive transfusion. And hypomagnesemia also will be there. What is not seen is hypokalemia will not be seen, and hyperkalemia will be seen. Okay, in massive blood transfusion. And coming to the 32nd question, the specific marker for alcoholic liver disease. Yes. So this is a very classical question. In alcoholic liver disease, very standard specific thing is if you estimate the levels of GGT, that is a gamma glutamyl transferase, that is a very, very specific enzyme. If this gamma glutamyl transferase enzyme is elevated, then that is actually tells that the alcoholic liver disease, alcohol mediated liver injury will cause a rise of this gamma glutamyl transferase. They are separately given SGOT and SGPT, so that the AST to ALT ratio, that significance they have not given here. They have given separately the enzymes. So GGT will be the preferred option. Okay. Then coming to the 33rd question, all of the following can cause non-immune hydrox except, yes. So all the things, say for example, thalassemia can cause hydrox fetalis, and parvovirus infection can cause destruction of the RBCs, can cause hydrox. Chromosomal abnormalities, yes, can cause a hydrox. But all these things are the non-immune hydrox. But what is the can cause your immune hydrox is nothing but your erythroblastosis fetalis, that RH incompatibility, that is something like an anti-D and a D antigen. So that reaction is actually an immunological mediated one. So Immune hydrops will be caused by our erythroblastosis fetalis, whereas thalassemia, parvovirus infection, chromosomal abnormalities will cause non-immune hydrops. Non-immune hydrops. So the correct option here is the erythroblastosis fetalis. Okay. Moving on to the 34th question, SRY gene is present on which chromosome? Yes, this is one of the very standard question. SRY gene is nothing but actually this is the sex determining region. Okay, SRY gene, sex determining region Y. This is actually a Y link and this gene is found only on the Y chromosome. If this gene is found only on the Y chromosome, and from the picture you can see that it is present on the short arm, that is in the P arm, P for petit. Yes, so short arm of the chromosome Y, this SRY gene is present. SRY gene is present. Okay. Moving on to the 35th question. Yes, so 35th question, a 45-year-old male presented with features of nephrotic syndrome. His creatinine value, they are given it as a 4.5. Yes, the creatinine value is for high. And which of the following statement is related to his condition? So we have to see what are all the true statements. Kidney biopsy is confirmatory in FSGS. Yes, FSGS, that is in the focal segmental glomerulosclerosis. If you take a kidney biopsies, and if you could see in the glomerulus only a segment alone, okay, only a part of the glomerulus, only a segment of the glomerulus alone, if it is involved, then it is actually a confirmatory for your FSGS. Yes, and biopsy is confirmatory in amyloidosis. Yes, to demonstrate renal amyloidosis, usually they will do biopsy. So biopsy, you can see demonstration of this amyloid in this glomerulus in the mesangium. So all these things is confirmatory for the amyloidosis. You demonstrate, you take biopsy, you do Congo, then you demonstrate amyloid. Yes, it is confirmatory for amyloidosis. Okay, and usually they will be giving you a 24-hour observation is essential of the kidney biopsy to monitor for any complications. Okay, and minor hematoma, minor hematuria is usually common during after kidney biopsies. And the hematoma is another such complications, separate complication which can occur after kidney biopsies. Okay, so only for hematoma alone, if it is present, hematuria will not occur. Hematuria can occur as a separate event. So other three options are right, like kidney biopsy confirmatory in FSGS, demonstration of the amyloid, and 24 hours essential for the kidney biopsy procedure. And then we have also one question. The South Indian lady was presented with a nasal mass. Identify the lesion. Yes, nasal mass, South Indian lady, you will be thinking of rhinosporidiosis. Rhinosporidiosis. And what you could see in the rhinosporidiosis, if it would have been a epithelium, and you can see the sporangium. Okay, these are the thick walled sporangium with all this small, tiny spores. This is the rhinosporidiosis. Rhinosporidiosis. But some students say that the examiner also given as a word like a Mikulicz cell and those things. If Mikulicz cell and all given, that it is then it is a case of rhinoscleroma. But if it is not given, then it will be a case of nasal mass and this similar kind of image with thick walled sporangium with the spores. Then it is a diagnosis of a rhinosporidiosis. Okay. So guys, these are the questions we have from the pathology end. And as you could see that from direct evidence, we have 36 questions actually from the pathology. So such an important thing for your exam. And we are very happy to say that out of these 36 questions, almost every questions actually came from a workbook. And I discussed with you with the screenshot, with the proof of evidence with the screenshot. It's a really happy moment and proud moment for all of us. And definitely, I expect all of you to score a good rank in the INICET exams. All the best, guys. Wish you all the best. Thank you.